I am recording now. Thank you so much, Andrea. Oh my God. So many buttons to push. I will record the beginning of that lecture as well. All right. Okay, so we understand that genetics is talking about heredity, transmitting traits one generation to the next. We're talking about parents transmitting it to offspring. The offspring would be F1, F2 for any further generations. Genetics is the study of that process. Characters are essentially like genes and alleles. And then traits are basically the phenotype, so the outward expression of that. All right. So I got a question. Awesome. Okay, so the questions, perfect. Should the F2 generation description say cross of two F1 generations? Yes, it should. Thank you. Let me highlight that. Why did it turn red? Good job. Thank you for catching that. Right? And then as for the difference between characters and traits, the difference between characters and traits, just a second, are basically the difference between genotype and phenotype, right? So for example, if I go back to that slide, we can have different versions of the same gene, right? Those are alleles. And then those, the combination of those expression is your phenotype, exactly what we observe. So whenever I use the term trait, that would be like observable characteristics, phenotype, whereas a character is going to be like genes, but technically alleles, which we'll define in a minute. Does that make sense? All right, perfect, good. All right, good, good, good. Okay, all right, so, okay, so excellent questions, keep them coming. All right, and observations, because if I need to change something, please let me know. All right, so in order to understand modern day genetics, we need to kind of understand what Mendel discovered through his experiments. So I'm not going to go through the pea plant experiment specifically because I do believe your reading assignment kind of goes through that. You may have heard it elsewhere, but what I do want to go through is the observations that Mendel made and the resulting hypotheses that he got from his experiments. So he came up with, I think there's 3 hypotheses. Let me see, 1, 2, 3. Okay, 4 hypotheses. Notice there's a clicker question. And I'm going to go through each of these. The first of these we've already sort of talked about, and that is that Mendel found that every basically character had different versions. So just like we talked about, and what we now understand is that this meant that every cell had different versions of the same gene. And it was because every cell had different versions of the same gene that an offspring could have different possibilities, right? It wasn't a blending of characteristics from the parent. Sometimes it could be something completely unexpected, like if you have 2 purple flowers and you breed them and they result in like a white flower. Like, where did that come from? So he discovered that even though you have a single gene, right, like flower color, every cell could have different versions of that gene. So again, what we now know to call that, different versions of the same gene, are called alleles. And these alleles are carried in the same spot on a chromosome. That location is called a loci. And those alleles are present on homologous chromosomes. So that's what you see in this picture, right? In this picture, you're seeing homologous chromosomes. This would be the same chromosome, let's say like chromosome 1. So all of these homologous chromosomes, these both contain all of the same genes, but they may not contain the same version of those genes. So for example, this picture is showing P color. So this chromosome and this chromosome, they're homologous. They have the same genes. One of those genes is including the gene for P color, right? And it's right there, same spot, same loci. However, there are different versions of the same gene. So the maternal chromosome carries the allele for yellow P color, whereas the paternal chromosome carries the allele for green P color. So same gene, P color, different version, which is an allele. Right. So that was his first hypothesis, right? And again, that was kind of generated from the fact that he was breeding flowers, like he would breed 2 purple flowers and would randomly get like a white flower. So that just showed that every cell has different versions of genes or alleles. His second hypothesis was through continuous breeding of these plants. And what he found is that for what we're kind of looking at, for each gene, an organism inherits 2 alleles. So that would be from each parent. Those are on homologous chromosomes. And like we talked about, what that means is that, again, I'm just going to highlight that because that's really the important part, is that every cell or every offspring would inherit 2 alleles and 1 from each parent. So first off, I want to mention the law of independent assortment. So that's at the bottom of your slide. And basically what the law of independent assortment says is that during meiosis, when crossing over is happening, and during cell division, specifically during mitosis, or I guess specifically during anaphase of mitosis, when the chromosomes are separating, it basically says that there's really no genes that travel together, or yeah, like chromosomes that travel together. So for example, if you have 2 populations of DNA, or like 2 cells, right, that are going to be divided, this cell will not receive all of the maternal chromosomes, and this cell will not receive all of the paternal chromosomes, right? It is completely random which chromosomes that cell will get during cell division, and also during crossing over, it's completely random which alleles will be moved around. So that is the law of independent assortment, that the assortment of chromosomes and therefore alleles is basically independent of each other. So as we will learn later, that is not necessarily accurate. What we know now is that there are some genes that are linked together, right? However, if a gene follows traditional Mendelian genetics, genes will travel independently of each other. And that is the law of independent assortment. So we'll go into that in a little bit further depth a little bit later. But the law of independent assortment just says that genes don't travel together. So I have a question. When the chromosome ends cross over, is it only the outer portion that crosses over, or do alleles in the middle of the chromosomes cross over as well? Excellent, excellent question. The answer is basically like during crossing over, even though it kind of looks like it's just the ends moving, during crossing over, they're so twisted and kind of like tangled up, any alleles can move around. So I think that picture that we looked at earlier of crossing over, where it was just like the tips, that was vastly oversimplified. The resulting chromosomes after crossing over are just like crazy amalgamation of alleles. Excellent question. Right. So, okay, so we understand the law of independent assortment. We know that every offspring inherits 2 alleles, one maternal, one paternal, because they're inheriting 2 copies of each chromosome, maternal and paternal. And what Mendel found is that certain traits or certain phenotypes were always expressed, whereas some traits were less expressed, or what he described as masked. So we'll get into that in a minute. And we'll talk about dominant and recessive. But first, what I want to talk about is how he described the kind of the fact that if you have two versions of a gene, right, you have two alleles, if your maternal and paternal chromosomes contain the same version, that is called homozygous, right? Remember, homos is Latin for the same. So you can see in the figure to the right, you can see on the far left of that figure, if you have a combination of chromosomes that can both contain the same gene, right, P color, but also the same allele, which is both alleles, the maternal and paternal alleles, both code for yellow Ps. Those alleles are the same, yellow Ps. So that is homozygous, so the same. And you can see that on the far right as well. You can see that that is a homozygous. Those are homozygous chromosomes, both containing the allele for green P color. So the alleles are the same, therefore they're homozygous. And then in the middle, you can see a combination where the maternal chromosome carries the allele for yellow Ps, and the paternal chromosome carries the allele for green Ps. So these are different versions of the same gene. Because the offspring inherited two different versions, we call that heterozygous. So hetero meaning different. So heterozygous chromosomes means they have different versions or different alleles. Again, of the same gene, so still talking about P color, but that organism has the alleles for both yellow and green. So question. So homozygous are always in the maternal or the paternal chromosomes. So homozygous is describing the combination the combination of alleles on both the maternal and paternal. So when you say a pair of chromosomes is homozygous, that means the maternal and the paternal both have the same allele, right? Does that make sense? Maybe a little bit? Okay, perfect. Right, so it's just talking about the alleles present on both of those, right? So homozygous is always going to be the same version, same alleles on both chromosomes. Heterozygous is just, all that is saying is that there's different versions on your two chromosomes. Perfect, good question. All right, so we understand Mendel's hypothesis. One is that every offspring inherits two chromosomes, one maternal, one paternal. They contain the same genes, but potentially different versions of the same gene. If they contain different versions of the same gene, that's called heterozygous, right, shown by the middle pairing of chromosomes. Or the offspring can inherit two chromosomes, maternal and paternal, but they could contain two versions of the same gene, but those versions are the same, right? So they're both yellow or they're both green, and that is homozygous. I have a question. Good, love it. Can horizontal gene transfer occur through this process, or is this all vertical? That's an excellent question. As far as we understand, yes, as far as we understand, in sexually reproducing organisms, which is what we're talking about with this type of genetics or this type of inheritance, horizontal gene transfer does not occur. So horizontal gene transfer pretty much just occurs in single-celled organisms like bacteria. Viruses can mediate this process as well. So it would not necessarily be directly involved in this process. So this would be strictly vertical gene transfer. So kind of inheritance from parents to offspring. Excellent question. I will say that we do know that within an organism, so yeah, within an organism, there are genes that can kind of move from chromosome to chromosome, but that is not necessarily included in inheritance so much as it is included in diversity, if that makes sense. It's pretty crazy. Good question. All right, so we know, like I mentioned, we're inheriting chromosomes with alleles. They could be the same allele, homozygous, or they could be two different alleles, heterozygous. So if we're looking at those chromosomes, how do we know, for example, with the heterozygous chromosomes, how do we know which trait, yellow or green, will be expressed? So that is Mendel's third hypothesis. And his third hypothesis basically determined that different alleles are expressed more than other alleles. So, for example, and he termed this basically dominant and recessive alleles. So whenever you hear the term a dominant allele, what that tells you is that if that allele is present in a combination of chromosomes, it will always be expressed, right? So dominant, it dominates, right? It is always expressed. Whereas recessive alleles are only expressed if the dominant allele is not present, right? So it's sort of like if you think about a classroom. Remember those days? If you think about a classroom and you think about like me standing at the front of the classroom, like yelling, not angrily, just because I'm a loud person, I'm a dominant presence in that room. I'm yelling, I'm loud, I'm taking over. And then you think about somebody in the crowd sort of whispering. They're very quiet. So who can you hear in that room? In that room with a person that's like yelling versus somebody that's whispering, the dominant voice will be heard. However, if I am all of a sudden like leave the room, like run out to use the bathroom or something, then only when that dominant presence is gone, then you'll hear that whispering, right? So that's kind of with dominant and recessive. If the dominant presence is there, it is expressed. If the only time the recessive allele will be expressed is if the dominant is absent. Okay. Again, we know now that there are exceptions to this rule, which we'll talk about later. I always love the exceptions to the rule. All right, so but let's talk about this first. This day called for like a Trenta cold brew, so that is my reusable straw. Don't judge me. Okay, so let's talk about it. Yes, soon, Starbucks. You can't go in, but you can drive through. Just use a lot of sanitizer. All right, so if you look at your chromosomes in this picture, this really explains kind of how dominant and recessive alleles are expressed. So this is mimicking what we saw on the previous slide, right? So on the left, we have a homozygous pairing. On the right, we have a homozygous pairing. And in the middle, we have heterozygous. So that's looking at this again. So on the far left, you see that both of the chromosomes contain the allele for yellow seeds. That would lead, I mean, we'd have no other options there. 2 yellow alleles will mean that we will get yellow seeds. All right. On the far right, you see a chromosome pairing. These are homozygous chromosomes, meaning they contain the same alleles. In this case, both chromosomes contain the green allele. So in this case, again, there's no other option. So if both alleles are coding for green pea color, you're going to get green peas. Okay, so let's focus on the one in the middle now, which is our heterozygous phenotype, or genotype, because we're talking about alleles. If we're talking about alleles, we're talking about genotype, different versions of the same gene. So if we look at that, our heterozygous genotype is that we have one chromosome with yellow allele and one chromosome with a green allele. And what you see is when you cross those two, or when those two are expressed together, what you get is yellow phenotype. What this tells you is that yellow is the dominant allele, because we're not seeing green. What this means is if yellow is present, that yellow will be expressed. Notice the only time we get green seeds is if the yellow allele is completely absent. Okay, so that's basically how we determine dominant versus recessive, is we have to ask, in a heterozygous pairing, which one is expressed? That's really the only way we can look at that. So if you look at this, we can also use words to kind of describe the genotype of these chromosomes. So on the far left, now we know that yellow is the dominant phenotype. And so we look at the genotype of these alleles, 2 alleles coding for yellow. Since we know that yellow is expressed in a heterozygous crossing, we know yellow is the dominant phenotype. So we would describe a pair of chromosomes that both contain the allele that codes for yellow peas as homozygous dominant. Both of those chromosomes contain the dominant allele. Right. On the far right, you're looking at a pairing of chromosomes that contain the same alleles, but we know from the heterozygous crossing that that green is the recessive phenotype because it was not expressed in the heterozygous crossing. So if we look at the far right, we see a homozygous, they contain the same allele, and that allele codes for green seed color. Since we know that green seed color is the recessive trait, we would say that these chromosomes are homozygous recessive. So I have a question. I'm going to get to it in just a second. So we have homozygous dominant. Both chromosomes contain the dominant allele. We have homozygous recessive. Both chromosomes contain the recessive allele. And then in the middle, we have heterozygous. So when you're describing a genotype, you have to say homozygous or heterozygous. If you say homozygous, you have to further explain, is the pairing of the same allele, are they dominant or recessive alleles? Okay, so the question is, can 2 dominant happen, or does it always come as dominant or recessive? So if you have homozygous dominant, that would be 2 dominant alleles present. Or homozygous recessive would be 2 recessive alleles present. Only heterozygous is when you have different versions. It's a good question. All right. So again, homozygous dominant, you have 2 of the dominant allele present. The dominant allele is going to be expressed. 2 yellow alleles leads to yellow seeds. In a homozygous recessive, both recessive alleles are present. You're going to get the recessive allele expressed. In this case, it's green. In a heterozygous, you have 2 different alleles. You have both the dominant and the recessive allele present. And if the dominant allele is present, it will be expressed. And that's why in the middle, when you have the heterozygous pair, you get the dominant allele expressed, which is the yellow seeds. So a lot of times, heterozygous pairs, we would describe those as hets, just H-A-T. Let me put that on here so that heterozygous so that's if you see me use that, or if you hear me use that, that's what I'm talking about. If you hear me say a het, I'm talking about an organism that has a pair of chromosomes with 2 versions of the same, or sorry, with both the dominant and recessive allele present. All right, another question, perfect. Do you know that an allele is dominant or recessive prior to pairing, or is it only known in heterozygous pairings? So you would, that's a good question. And basically, you have to do heterozygous pairings to figure out what the dominant or recessive allele is. So we would only know that yellow seed color and green seed color are dominant or recessive respectively by doing a crossing of two hets and seeing which one is expressed, or basically just looking at a het and seeing which one is expressed. So before Mendel's experiment, we didn't know that yellow peas were dominant and green peas were recessive. He had to do all these crossings to figure that out. So, good question. All right, so that's his third hypothesis. So going through that again, every organism gets two chromosomes. They contain different versions of the same gene or alleles. His second is that you can have two versions of the same allele. You're homozygous, or different version, different alleles. That's heterozygous. Law of independent assortment says those are not traveling together. They're inherited kind of independently of each other. His third hypothesis is that alleles have different expression patterns. They can be always expressed, in which case they're dominant, or they can only be expressed if it's the only allele present, basically, which would be recessive. So recessive alleles are only expressed if the dominant allele is not there in traditional Mendelian genetics. Right. The fourth hypothesis kind of really goes with the law of independent assortment in how these traits are inherited. And the fourth hypothesis is, I think I talked about it when we talked about law of independent assortment, actually, is what's called the law of segregation. And that basically, the law of segregation says that when sperm or eggs are generated via meiosis, it is completely random which sperm or egg when a sperm or egg are generated, when you generate those haploid cells via meiosis, it is completely random which of those cells, which of those 4 sperm cells, for example, will receive which chromosomes. So basically, which alleles, right? So it's not like this picture doesn't show it very well because the show is sort of not showing that. But you can see that the way this figure is showing it is that you have the maternal and paternal in your diploid organism, different alleles, and via those 2 processes of cell division, if it's just basically like those 4 haploid cells can receive maternal or paternal chromosomes, is basically what that is saying. Because of crossing over, if you look at the right side of the figure, you see the 2 haploid cells that have the blue chromosome, and then on the left you see the 2 haploid cells that contain the red chromosome. Because of crossing over, the 2 red chromosomes do not necessarily contain all of the alleles from the original maternal chromosome. And then the two blue chromosomes do not necessarily contain all of the alleles from the original paternal chromosome. So crossing over really mixes up alleles, and then how those alleles are divided into haploid cells. Basically, this is again just saying that this genetic information doesn't necessarily travel together. Does that make sense? It's a little bit hard to describe. But that's separation, so you're not necessarily going to always see paternal alleles in one cell. You'll have a mix of maternal and paternal, or you could have 2 paternally inherited alleles, right? So this is why I have some alleles that I may have some alleles that came from my mother and came from my father. I may have some alleles that came from my father only. I may have some alleles that came from my mother only, because those alleles via crossing over are really mixed up. Does that make sense? Sort of? A little bit? Okay, good. I have a question. Would all egg or sperm from a single individual have the same alleles? No. That's kind of what this is talking about, is that when an individual produces, that's an excellent question, and a way better way to say what I'm trying to say, so thank you, is that basically what this says is that every sperm carries a different genotype, and every egg carries a different genotype. This really helps explain why if you have a sibling, you don't look exactly alike, because you are generated from a sperm and an egg combining. That sperm and that egg has a completely different genotype than the sperm and the egg that come together to form your sibling. So this is why siblings don't look exactly alike, why siblings have different genotypes, etc. Excellent way. Thank you, Noelle, for putting that into words that I was struggling with. All right. Do we feel okay with that? Law of segregation. That says that sperm and eggs basically have different genotypes. Is an excellent way to say that. All right. Okay, so we know the 4 hypotheses. We understand that there are different alleles for every gene, that alleles can be, or that an individual can be homozygous for those alleles or heterozygous for those alleles, that the combination of those alleles, and then different alleles can be dominant or recessive, and then again, via meiosis, sexual reproduction, etc. This means different combination of genes are found in all of the haploid cells generated via meiosis. Goodness gracious. All right. So those are the 4 hypotheses. So now what we need to know, well, let's do a quick clicker question. I'm going to give you the answer, but I want you to see these regardless. All right. So blank would mean that 2 members of an allelic pair, so right, 2 chromosomes containing alleles in a zygote would be the same. So which of those terms would indicate that the alleles are the same? Go ahead and type it in the chat box. Oh, yes, looking good. I see a lot of D's, and that is the correct answer, as you can see here. Excellent, good job, right? So homozygous would mean that 2 alleles are the same. All right, whoop, I guess that's the only clicker question I had, so good job. All right, so homozygous means that 2 alleles are the same. Okay, so this is talking sort of about how we can apply this information. And again, we understand that there are different alleles. We understand that there are dominant and recessive alleles. But what we need to more understand is how we can sort of predict the likelihood that a certain allele will be expressed when a cross happens. So that's really what Mendel was doing. He was trying to figure out the pattern of inheritance, basically dominant versus recessive, and which alleles would be expressed, and likelihood they would be expressed. And that's what he was doing through his experiments. So, as you are probably aware, this means we're gonna talk about Punnett squares. Yay! So, in order to understand Punnett squares, sorry, you probably saw that get bigger. In order to talk about Punnett squares, first, we can look at the right side of the slide on the top. And what you're looking at here is the P generation. So again, we're talking about Mendel's experiment. And what he did is he generated these plants that were what he called true breeding plants. And what that means is basically he bred purple plants together over and over and over and over and over until when he bred 2 purple plants together, there was never any white offspring. All right, so that was true breeding. No matter what, when you cross them, they generated purple plants. So this would mean that they were homozygous for the purple allele. Right. And then he also did the same thing for the white flowers on these plants. So we bred white flowers together over and over and over and over and over until all of the offspring that ever resulted from crossing two white plants together were white. So he knew that those flowers would have had to have been homozygous for the allele coding for white flowers, right? So we have true breeding purple flowers, homozygous for purple allele, and we have true breeding white flowers, homozygous for the white allele. So the question is, which of those is dominant? So you can figure that out, and you can see on the P generation at the very top, he bred the purple and he bred the white flowers together. And what he found is that all of the offspring of purple and white breeding were purple. So this would tell you that that means that purple is the dominant allele. So you take purple and white, you breed them, you get all purple. So that means that any of the genes from that purple flower are dominant, right? So that's the dominant allele. It masks the white allele, and therefore the white allele is the recessive allele. Another thing is if you cross, so I guess what that means is that we can designate the purple allele as the dominant allele, and we can designate the white allele as the recessive allele. So if you look at that top panel, you can see how we typically do that. We describe a dominant allele using capital letters. So for example, since we know that the purple flowers, or purple is the dominant phenotype, if an organism is homozygous dominant, that means two alleles that are present are both the dominant allele. So you can see in this case, if we're talking about flower color, that would be capital P, capital P. So capital P would be homozygous dominant. And then because we know that the white allele is recessive, and because we know that these true breeding white flowers had both recessive alleles present, we describe the recessive allele as being lowercase p, so homozygous recessive would be lowercase p, lowercase p. So if we cross a homozygous dominant with a homozygous recessive, all offspring will contain a dominant allele and a recessive allele. So we can talk about how we know that later. But that's what we're looking at here in the F1 generation. We know that all offspring have a dominant allele and a recessive allele, so they would be heterozygous, right? Dominant and recessive alleles are both present. That's a definition of a heterozygous that we've talked about so far. So the question there is, what is going to happen if we cross or breed two heterozygous individuals, or two hets? So if we cross a heterozygous capital P lowercase p with another capital P lowercase p. So the F1 generation is hets, and what we're trying to look for is what that would mean for the F2 generation. So we can complete a Punnett square to do this, and that's what the bottom of this panel is showing. So in order to set up a Punnett square, you see that you basically just draw a quadrant. You see a cross in the middle, and what you put on the left column, or well, I guess what you will do is you'll separate the genotype across the top and across the side, and then we'll pair the different alleles to kind of look at the likelihood or expected genotypic ratio. And from the genotypic ratio, we can predict a phenotypic ratio. So we'll talk about that in a minute. So basically, we just set up a quad grid. Along the top, we put one of the genotypes. It doesn't matter which genotype is put on the top or the side. All that matters is that one column, above one column, you have one allele, right? So we're doing a heterozygous individual. Every individual has two alleles. You are separating these alleles on columns, so, or rows. So the first column has the first allele, right, capital P. The second column has the second allele, lowercase p. All right, so that's on the top. On the side, you're going to put the other genotype. So again, in this case, we're crossing PP times PP. Sounds ridiculous. let me write that, insert text box. So here we're doing this. So you can see we put the first genotype along the top, we put the second genotype along the side, as opposed to putting different alleles on different columns or putting different alleles on different rows. So capital P, lowercase p, capital P, lowercase p. And what we do is we just work to combine those. So in the first square, we have the dominant allele and the dominant allele. So this gives us a homozygous dominant genotype. In the second square, we have lowercase p, so the recessive allele, and we have a dominant allele coming from the left. That would give us a heterozygous genotype. Moving down, capital P, lowercase p, heterozygous, and then in the last square, we have the recessive allele and the recessive allele. So that's giving us a homozygous recessive genotype. So from this pairing, from this Punnett square, we can establish what our genotypic ratio is. We have, from this pairing, we have 4 possible outcomes, right? One of those is a homozygous dominant. So you'll always write that first, homozygous dominant. It's 1 out of 4 likelihood that we will have a homozygous dominant genotype. 2 of these actually have the heterozygous possibility. So 2 out of 4, or half, of the offspring, it's likely that they will be hets. And then it's a 1 out of 4 likelihood that the offspring will be homozygous recessive, shown lowercase p over here. So our genotypic ratio would be 1 to 2 to 1. And that's talking about the fact that we have the possibility of 1 homozygous dominant, 2 heterozygotes, and 1 homozygous recessive. That's our genotype, so it's just talking about the alleles present. However, we have to interpret this further to talk about, okay, those are our likely genotypes, combinations of alleles, but what does this mean for the observable characteristics? So that's also shown in this Punnett square to the right, but we can also look at that by just looking at the genotypic ratio. If we have capital P, right, that's the only trait that's present is the dominant trait. So that means the dominant trait will be expressed. And in this case, that's a purple flower. With our heterozygote, we have the dominant trait and the recessive trait, or allele present. If the dominant allele is present, the dominant allele will be expressed. So we have 2 possibilities where they're hets. That means, again, both of those possibilities would result in purple flowers. So that means that the 1 homozygous dominant plus the 2 heterozygote individuals, both of those would result in purple flowers. So far we have 3 out of 4 possibilities for purple flowers. And then we also look at our genotypic ratio. We only had 1 out of 4 possibilities where both alleles would be recessive or homozygous recessive. There is no dominant trait present. That's the only time we would get the recessive allele expressed. So again, that's the likelihood is that 3 to 1, you're going to get purple flowers as opposed to white flowers. Right. Does that make sense? Throw me a yes or I'm not really sure in the text box. Okay, perfect. All right. Perfect. Thank you so much. Okay, so we understand genotypic ratio as opposed to phenotypic ratio, but you have to have the genotypic ratio in order to predict the phenotypic ratio. That's basically how it's going to help you determine that. Could I repeat F1? Could I repeat F1? Up here? So basically what F1 is it's just we cross the true breeding or homozygous dominant genotype with the homozygous recessive phenotype, and we generated the F1 generation. And if you do the Punnett square for this, so right, if we did a Punnett square crossing the homozygous dominant versus the homozygous recessive, what you would find is that all 4 possibilities would be heterozygotes. So I don't know why this figure didn't show the Punnett square for this, but that's how we know that if we have the true breeding purple flowers, capital P, versus the true breeding white flowers, lowercase p, when we breed those, all of those would be heterozygous, or capital P, lowercase p. Does that make sense? Yes, of course. All right. So what I want to mention is in this Punnett square we just did, we were talking about one trait. And that trait was flower color. If you are talking about one trait, that is called a monohybrid cross. And that's why I have this down here, right? Just circled it so you would see what I'm talking about. Monohybrid cross, that mono means one, and what it's talking about is one trait. So, unfortunately, we can also use Punnett squares to look at inheritance patterns of more than one trait. And in this case, we're going to talk about a dihybrid cross, which is talking about di meaning two, two traits. Why would we do this? That's a question I ask myself every day, like, why would you do this? But the answer is, as we will talk about later, we talked about the fact that due to the law of independent assortment and due to the law of segregation, Mendel, if a gene follows traditional Mendelian genetics, traits are inherited, are independent of each other, right? So, for example, if you're looking in this experiment outlined on this slide, you're looking at two traits. On the left, you see a pea that is round and yellow. That's two different traits, basically like shape or texture or whatever, round, and color, yellow. And on the right, you see a different seed or pea that is green, so a different color, and has a different texture or shape, which is wrinkled. So you're looking at two traits, color, yellow versus green, and shape or texture, which is round versus wrinkled. So if we're talking about traditional Mendelian genetics, due to the law of independent assortment and segregation, that would say that when you cross the organisms with these genes, the traits of round and yellow or wrinkled and green are not inherited together. So for example, you could have yellow peas that were wrinkled, all right? So they're not inherited strictly together. We now know that that is not the case for all traits. So some traits are inherited together, and these are called linked genes. And we'll talk more about these later, but I'll give you like one example. We know that the alleles coding for like red hair and freckles are linked. So it is very, very, very rare to see an individual that has red hair and no freckles. Freckles, you can have freckles without having red hair, but specifically, like if there is an allele for red hair present, it typically attracts the allele for freckles, right? So it's very rare to see those traits kind of inherited separately. Those are linked genes. So the reason we do a dihybrid cross is basically to ask ourselves, are these traits linked? Do they travel together? Or are they inherited independently one another? Do they follow kind of Mendel's hypotheses? So that's why we do it. Okay, so let's do one of these to discover how we can find that out. So this is kind of setting up Mendel's experiment, how he sort of figured this out. So we're starting with our pea generation, and we have true breeding pea plants. What Mendel found out is what I've outlined here on the left, is he found that the dominant allele, or capital R, codes for these round P's, and that the dominant allele for color would be yellow. So yellow would be our dominant color. I have a question. So it basically demonstrates to us if there's a relationship between different allelic pairs. Yes, it basically demonstrates whether they are linked or not. Excellent, good job. Yeah, perfect, thank you. So we're looking at here, again, two traits. We're looking at round or wrinkled and yellow versus green. So round is the dominant trait, wrinkled is the recessive trait, yellow is the dominant color, and green is the recessive color. So he figured that out. We know that, all right? So round and wrinkled would be two different traits, but since both of those are dominant traits, this would be an example of a P that is basically homozygous dominant for both of these traits. And then green and wrinkled, because we know both of those are recessive traits, if we had a homozygous recessive for both of those traits, it would be green and wrinkled, shown here. So that's why we know that this is the genotype for this phenotype, and this is the genotype, well, for this true breeding phenotype. So we cross these individuals, and as we talked about, so this does not do the Punnett Square, but just as we found out from this experiment, when you cross true breeding organisms, so homozygous versus homozygous, what you're going to get is a bunch of heterozygous individuals. So that's why we know that when we cross these homozygous dominant versus homozygous recessive individuals, what we get is heterozygous individual. So the F1 generation is a het for the seed shape or texture. So it has the dominant and the recessive alleles. And it is a het for the color. So it has the dominant allele, yellow, and the recessive allele, green. So what we want to know is if we cross two hets, what would be the phenotypic ratio? What would the phenotypic ratio be? Right. So we have a question. Is the P generation always a cross between two homozygous genotypes? No. Good question. So these, the P and the F1 generation that we're talking about in these slides are based on Mendel's experiments to kind of talk about his experiments. The P generation, the F1 generation, F2 generation will all depend on your experiment. So, but in Mendel's experiment, because he did all the true breeding stuff, and this was just to figure out kind of different patterns of inheritance, we're referring to P, F1, and F2 specifically coming from his experiments. It's an excellent question. Yeah. All right. Okay, so we have our crossing. What we want to do on the next slide is we want to look at a crossing between two individuals from the F1 generation. So what would happen if we crossed a het for wrinkled or smooth and a het for green and yellow, right? The reason we're doing this is because based on how the hets, the outcome of the het crossing, we can determine whether these genes have any relationship to each other, like Jennifer said. So are they linked or are they inherited independently? So let's look at it. So this is a lot. Let's break it down. All right. So the first question should be, how do we decide what is on each column or row? Right. So you can see we're crossing two individuals. Basically here we're crossing sperm versus egg. Right. We're doing hets. So capital R, lowercase r, capital Y, lowercase y versus the exact same. And what we want to know is the phenotypic or the genotypic ratio. And in this case, actually, we're looking at the phenotypic ratio. So let's not get there, right? I copied this over. We know the dominant is round yellow. The recessive is wrinkled green. Our genotype is capital R, lowercase r, capital Y, lowercase y. So when we're looking at a crossing, how we can determine what goes on the top of a dihybrid cross Punnett square is you can use the like anagram, I can't remember what it's called, but it's basically FOIL, which stands for first, inner, outer, last. Okay, so what that refers to is the boxes. So to set this up, you're going to have 16 squares, right? So we have 4 in the first column, 4 in the second column, 4 in the third, 4 in the fourth. You'll notice there's 4 boxes also in each row. So that's a total of 16. And this allows us to have 4 haploid genotypes on top of each column or row. So again, haploid cells would be the sperm and the egg. So that would be, we have 4 possible sperm from this individual, 4 possible eggs from this individual. Okay, so how do we determine what goes on top of this column? We do FOIL. So FOIL is looking at just one of these, right? So let's say the sperm is coming from this individual. So we do FOIL. First would be capital R of the round or wrinkled genotype. So capital R versus, and the first, how do I select both of them? There we go. First would be the first allele in each of those genotypes. So capital R, capital Y. First allele. Capital R, capital Y. Inner would be the inner 2. Inner would be lowercase r, capital Y. And that's what you see is on the second box. Outer would be capital R, lowercase y. And that's what you see on the third box. And last would be lowercase r, lowercase y, which is what you see on the last box there. So again, first, the first allele in each of these, first, outer, first, inner, oh my God, I'm sorry, here, outer, last. That's how we're getting what to put on top of each of the columns. You could do file or foil. It doesn't matter. The like what I just described would be file. It's however you can do it, just as long as you get all 4 possibilities and that they're independent of each other, right? We just use foil because that allows us to kind of take us through, but it could be file, however you want to do it. All right, so again, first, right, inner, outer, last. All right, so we did that along the top. You can also do it along the side. In this case, you're using the second individual, but because the second individual is exactly the same as the first, it's going to have the same combination. So along here, we do the same thing. First, outer, inner, last. So we have our first, which notice, our first row, capital R, capital Y. Inner, lowercase r, capital Y. That's our second one. Outer, capital R, lowercase y, here, and last. Woohoo! Okay, so that's just setting it up, but then you just go through and pair them. So with this one, we have a capital R, capital Y, capital Y. That gives us homozygous dominant for R, homozygous dominant for Y. Our second is lowercase r, capital R. We pretty much always write the dominant allele first, which is why we don't have lowercase r, capital R, but honestly it doesn't matter as long as you know what you're talking about. So here the result is 1 dominant, 1 allele for R, and then we have a capital Y, so homozygous dominant for color. And then you just do that box by box by box by box. So typically what you would see is if you do a dihybrid cross, we wouldn't have the phenotype along with the genotype. But your textbook tried to make this simplified, which is honestly like very nice. But we can look at this, and if we know that we have a genotype of capital R, capital Y, capital Y, capital R is homozygous dominant for round seeds. So that means that the dominant allele will be expressed. So we have round seeds. It's also homozygous dominant for color, which would be the Y, which would mean we have yellow, right? The dominant allele is expressed. If we go over here, right, so let's look at this one, lowercase r, capital Y, due to this pairing right here. We have two, this is a homozygous recessive for texture or shape. If we have homozygous recessive, that means the recessive allele will be expressed, which is why we have wrinkled seeds. We also have homozygous dominant for seed color, right? That means we have yellow color. So we can do that. What we would need to do, again, is go through each of these and look at each of those different phenotypes. So if we think about this pairing, right, if we talk about having dominant and recessive, we have actually notice four phenotypic, that's what it's looking at when we look at results. We have four phenotypic possibilities. We can have the dominant color expressed, which is yellow, as well as the dominant texture, which is round, so dominant. We could have the dominant texture, round, with the recessive color, so that'd be round and green. We can have the dominant color, which is yellow, with the recessive texture, which is wrinkled. Or we could have both recessive alleles, recessive color, green, recessive texture, wrinkled. So those would be our four phenotypic possibilities. And what you would do to find the phenotypic ratio is you would say, okay, how many out of this crossing result in yellow and round seeds? And you can see we have 1, 2, 3, 4, 5, 6, 7, 8, 9. So that would give us 9 out of 16 would be our phenotypic ratio for the homozygous dominant traits to be expressed. For our two heterozygous possibilities, you'll notice you have 3 green and round, and 3 yellow and wrinkled. And then we only have 1 outcome that would result in both recessive alleles being expressed, which would be 1 out of 16. So real quickly, I know we only have a minute left, but I just want to point out that if a dihybrid cross is indicative of Mendelian genetics, which in this case we know it is, the phenotypic ratio will be 9 to 3 to 3 to 1. That ratio is really important because in a dihybrid cross, if you get that ratio of offspring, that is a Mendelian inheritance pattern. If you do the cross and you get a different outcome, phenotypic ratio outcome, that means that it did not follow traditional Mendelian genetics. Okay. So we're out of time today, but that's okay because we got through dihybrid crosses, which is a big pain in the butt. When we come back on Tuesday for lecture at the same time, we will continue. So we know the basics of Mendelian genetics, and when we come back, we'll talk about the fun stuff. So what happens when it gets a little bit different. Okay, so if you have questions, I'll hang out on Zoom for a couple more minutes. You can type them into the chat and I will answer them. Also remember that you have the discussion board. Please post your initial post as well as your reply by Sunday at 11:59 p.m. You also have labs due by Sunday at 11:59 p.m. I pushed that back because of the crazy circumstances. You also have a Learn Smart reading assignment. That's a lot due by Sunday. Don't wait to the last minute. But do what you can do, right? If you're struggling, reach out. I am here. I will be here. Yeah. All right. So if you have questions, again, I'll hang out for a second. But if you need to go, that's totally fine, right? Okay. I'm trying to stop this. There we go. All right. See you later if you're leaving. If you want to hang out and ask questions, go ahead. You're welcome. Thank you. I miss you guys, and I wish you health. Bye. Yeah. Question, Talmud, what's up? If you want to, you can unmute your mic, or you can quick it in. Okay. Would you prefer correspondence via email, or you just want? Oh no, email me. That's fine. It might take me a little while to get back to you, but email is fantastic. Okay. You're good. Yeah, see you later. Have a good weekend. All right, if there's no other questions, I will be having open office hours. Open means that anybody can join, so there may be more than one person in the room, but I can always request it to be more personal. Oh yeah, Anil, for sure. Okay, so what I'm gonna do for everybody still in the room is I'm gonna end this session. I'm gonna start a new one, Anil Hangout, and if everybody else could just peace out. If you want to come to open office hours. Oh, a question really fast, and then Anil will start those office hours. Samantha, is the homozygous chromosomes, how do they become that? Is it after they duplicate for cell division? So remember that so you have a sperm and an egg, each containing a chromosome. When those combine, you get a zygote, and it has two chromosomes, two copies of each chromosome. So those are homologous chromosomes. If you have a homozygous chromosome, both of the chromosomes inherited from the mother and father would have the same version of the gene, right? So like green alleles, right? You can have homologous chromosomes inherited that are heterozygous for a trait, meaning they have different versions of the same gene. So homozygous is strictly talking about just if you have two chromosomes, are the alleles present on the chromosomes the same, or are they different, in which case that would be a heterozygous. Does that make sense? That's a good question. This is where it gets a little bit confusing because of the homologous and homozygous, all that terminology. So homozygous is both alleles are the same. So alleles are just basically genes, right? If you have the same alleles of a gene, right, green and green, that's homozygous. Heterozygous would be different versions. So it would be the same same gene, like pea color, different alleles, green and yellow. Oh, good. And if you have questions, again, this is like why that homologous, homozygous, all of those terms can get really confusing. So if you have questions, always reach out. All right, so I'm going to close down this session. I'm going to open a different session, Anil, and if everybody could just leave this room so that I can use this room for just office hours. I'd appreciate it. Thank you so much. I will see you soon.